Welcome back, everyone, to the OIS Retina Podcast. This is Firas Rahal here in Los Angeles. I'm part of the Retina Vitreous Associates here, and of course, I'm also part of Excite Ventures out of New York City, the host. And I'm delighted to have my friend Brian Levy as our guest today. He's going to tell us about his current ventures with InflammX. We'll get to that in some detail later, but also his history, which is thorough and long in ophthalmology. Brian's been an ophthalmology researcher and product developer and business manager, and he himself is an optometrist by training. So welcome, Brian. Thank you for joining us.
Thanks, Firas. Pleasure to be here. I can tell you that much.
I like the pictures in the background, by the way. Did you take those?
Yeah, my passion is wildlife photography.
Okay, we already learned something about you I didn't know. Maybe we'll come back to that. Those are fascinating and beautiful.
Yeah, I'm in Africa every year because I was born there and raised there. So I've been doing this since I was 16.
Those look like fairly close pictures. I'm already nervous for you. I think you're going to have to reconsider this part of your career. You're getting slower afoot, Brian.
I know. That's why I've got bigger lenses.
Very good. Absolutely worthwhile. Let's start with the history. Start all the way, your educational background, where you were trained and so forth, and the early part of your professional career, if you don't mind. I always find that very interesting.
Sure. So I was born and raised in South Africa, and I was educated there as well. I actually went to vet school first and then decided that I didn't want to be a vet, and ended up getting into UC Berkeley, the optometry program. So they gave me full credits for everything, and I only had to do 2 years to get an OD. So that was my first degree really coming out of something that I thought was useful. And following that, I actually had to go back to South Africa, and I had to spend 2 years in the military because I was conscripted. And if I didn't go back from the US, I would never have been allowed to come back into South Africa. And my family was there, and I've got a brother and sister still there. So I did. I went back, did my 2 years, and then went to Canada because I got offered a position at the University of Waterloo to do a master's program. And I did a Master of Science program at the University of Waterloo in comparative anatomy and physiology of the eye, which we can talk about another time, but that was probably my most fascinating 2 years that I can ever remember at school. It was fantastic. And I learned a lot. I learned a lot about eyes and about animal eyes, which is part of my interest, and came away with a master's and went into practice for about 5 years or 6 years in Toronto, and then got offered a position at what was Pacific Presbyterian Medical Center by Bob Stamper. I actually met him at Arvo, and he said to me, I've been reading the stuff you've been doing and publishing, and he said, I'd really like you to consider joining our department. And at the same time, I'd taken a course at NIH called Epidemiology and Biostatistics in Vision Research. And that actually really pivoted me into doing clinical research. I realized the importance of being a clinician and a researcher and how valuable that could be. And so Bob recognized that and asked me to come to San Francisco. So I picked up my family and off we went, my wife and 3 kids, and off we went to San Francisco. So I spent 6 years, Pacific Presbyterian became California Pacific Medical Center. Spent 6 years there. We did a lot of research there, particularly looking at new products coming from pharmaceutical and device companies. And, it also brought money into the department. So it was a win-win for everybody, but still saw patients there. And then I got recruited by Bausch Lomb. And so I thought, okay, do I want to pick up my family again and move to Rochester, New York? And they convinced me that, yes, I should do that, and I did. So we went out to Rochester, New York, and spent 15 years there. I came into B&L as the director of clinical development for devices, and ended up as a VP of R&D eventually for both device and pharma products, and then ultimately became the chief medical officer of the company. So went into the C-suite. And so that's what really taught me what goes on really in the corporate world. And also found that I had a pretty good knack in terms of developing products. So I got 3 products developed at Bausch Lomb, submitted 3 pharmaceutical products, that is. Retisert was the first one, and then Xylet was the second one, and Bezivance was the third one. So 3 NDAs, all approved and all commercialized. So that was, in 15 years, a pretty good, part of my life. And I enjoyed every minute of it. I have to say, B&L was a very interesting company, understood the ups and the downs, went through multiple CEOs, but learned every time something new happened. And you asked about Retisert. So Retisert came to us as part of an acquisition, and it was developed by pretty good drug delivery developer, Paul Ashton. And he had a company, and we acquired the company and the technology, and we basically developed it for non-infectious posterior uveitis. And this was an implant with steroid, acetonide, and basically the steroid would elute out of this implant for 3 years, so 36 months. But it was surgically implanted. So we had to implant it in the pars plana, suture it to the sclera, and basically leave it there for a 3-year period until it ran out, and then it would have to be replaced. And there were other products actually being developed at the same time. Ozurdex was one of them. And, that was a biodegradable. Ours was not biodegradable, but it did last a long time. So we went into the eye once. Now, everybody said, well, what happens when you put a steroid into the eye for 3 years? I mean, people are going to get cataracts and glaucoma and all of this stuff. And that is true. I mean, just about everybody got a cataract if they didn't have one initially from the uveitis anyway. But cataracts were removed, and patients actually got, really good vision with the resolution of the posterior uveitis. The macular edema disappeared, and vision was returned to these patients. And more importantly, they also managed, these were serious uveitis patients that had systemic disease and were on systemic steroids, a lot of times to control the uveitis. And so they managed to get to either a lower dose or off their steroids systemically. So it was a really interesting product, and it was quite successful. And I then went on to develop, the other products, Xylet, which was Lotamax, and an antibiotic, and then the Bezivance, which was a 4th-generation fluoroquinolone. The interesting thing about Lotamax was that was Bausch Lomb's first pharma product, and it was one that I was one of the investigators before I joined B&L at California Pacific Medical Center. We were running the clinical study, and we were part of it. So it was kind of a nice thing that I came to B&L, and there was the product that I'd worked on, and they were now selling it. So that was my time at Bausch Lomb. The company then went private. Our CEO decided to take it private. And so I left B&L and I moved to New York City. And in New York City, I joined a company in New Jersey called Aerie Pharmaceuticals. And at Aerie, I joined them as the chief medical officer. And there we developed the 2 Rho kinase inhibitors for lowering intraocular pressure. Rhopressa was the one, which is a monotherapy, and then the combination with latanoprost called Rocklatan. Again, both of those were approved and are now commercialized. And, long after I left, Alcon actually acquired the company. So, in terms of pharma, I have basically worked and got approved, 5 different pharmaceutical products, all successful.
Great products. I've used all of them, actually. You mentioned so many that we use commonly in clinical practice. I want to go back for a minute to the Retisert because I'll talk a little bit about the drug delivery subsequently, but as a backdrop to that, I remember when that came about, and these patients were on powerful systemic drugs or systemic steroids or both, and would get very sick, obviously, from the toxicities of these drugs. So we were using various angles on local therapy, retrobulbar steroids, ocular steroids were starting to get popularized with injections. We all thought, and it was a great development and an advance. What do you remember about its development? It was one of the first ones. Of course, Vitrasert predated that for CMV retinitis. I put a lot of those in the 90s and early 2000s. What do you remember about the development plan and the execution? Was it super challenging? What were the barriers there since you were one of the first ones?
It was super challenging. And what were the barriers? Everything. I learned everything about drug development when developing Retisert. It was such a complex project. And also dealing with another company, a private company that we had acquired, how to deal with the people in the other company as well, because they had a lot to do with us. They were, we had to get manufacturing done and then transfer manufacturing from them into our sites. And so it wasn't just developing the drug, but everything that could go wrong went wrong. And that's how you learn. And so along the way, there were mistakes from everybody along the way. Nobody had done this before. This was really the first steroid implant that had been approved by the FDA. And I have to say, we got a lot of help from the FDA. And it's also the one thing I learned is you have to talk to the regulatory authorities on an ongoing basis as things happen. Like, at one point there was a stability failure in the stability program. And this is not that easy that you can say, okay, stop shipping the drug. The drug is sitting in people's eyes. So I called Wiley and I said to him, what do you think we should do? Do I have to take them all out? He said, no. He said, just follow them, follow the IOP, follow, that the steroid is still working as it should in terms of treating these patients, get input from the investigators, and then let me know when the stability failure was over. But, it also taught me, having conversations with the regulatory agencies is absolutely critically important in terms of drug development. But I also learned that, drug development is not for the faint of heart. It's a roller coaster ride. Things go well and they go badly. But at the end of the day, my motto has always been to do well by doing good. And I believe so far I've met that criteria.
You totally have. And the pitfalls are anywhere from the very beginning even to the very end. My friend Ron Polanski mentions this all the time. The risk is there throughout, even post-FDA approval, and we've seen this with drugs recently in the last 5 or 10 years in our space that have been commercialized and then had more than a bump in the road and didn't succeed commercially for various reasons. Retisert was a brilliant product, and I used it again. I sewed it into many posterior uveitis cases with good results. What do you think transpired then commercially for them? Was the onset of in-office steroids, which is a different way, an easier way potentially to put these things in? Is that why that sort of faded away, or do you think there were other reasons? It was mostly the in-office part of it.
No, I think that was definitely part of the reason. I mean, clearly, this was a more complex system. It was more complex for the doc, but it was also more complex for the patient. And there were serious, side effects. There was, the cataracts and then glaucoma. A lot of patients had to have trabeculectomies. Interestingly, I got a call one day from one of the glaucoma guys. He said, I don't know what you guys have done. He said, but these are the trabeculectomies that I dream of. And because
they have steroid on board.
the steroid was
Yeah.
on board, they had the best trabeculectomies.
Yeah.
But it's a challenge to everybody. And then, when the bioerodible came along, I think that did change the dynamic. Although there was still the smaller implants that Paul Ashton went on to develop, which I think that went to Alameda, and I think those work pretty well. I think they still do. And that one did not require the suturing, but again, they're not bioerodible, so they're sitting inside the eye. So I think having bioerodible drug delivery, depending on the indication, is definitely the way to go. And obviously, the longer one can get, with it, there's obviously a small, volume that one has to work with, but as large as the eye can take it and the drug can deliver for longer and longer, the better off it would be.
It's a good segue. You and I are both on the SAB at Rivana, a company bringing some of these products to the world right now. What are your thoughts about biologics now, the new frontier? These have all been small molecule steroids. We've gotten pretty good about delivering that in a longer acting fashion, as you just mentioned. What do you see? It's obviously a challenge, but how big a challenge is getting drug delivery, let's say in office especially, but in any venue, into the back of the eye with biologics? You're an expert in this, and I would love to hear your take.
Well, I think biologics are more complex, period. And, doing drug delivery with a biologic, as we've seen, has been a huge challenge. I mean, everybody would like to have a drug delivery system for the anti-VEGFs, and nobody has, as yet, really succeeded. And that doesn't mean that, people haven't tried. But I think as we've learned more and more about the nature of the biologics, the not just the chemical nature, but the ability for the biologics to be placed in the eye and delivered for a long period of time, I think is starting to see a glimmer of success. So I do see that as something that in the not too distant future is going to be achieved. And I think that's going to be, significant benefit for patients in particular, but also for the logistics of, multiple injections and chronic injections to patients with macular degeneration or, diabetic retinopathy. So, and other potential diseases as well. But I think having drug delivery for biologics is the way to go. And from what I've seen through Rivana and what I've read and seen, presented from other companies, it does seem like we're seeing the light at the end of the tunnel.
I agree. It will most definitely be a game changer for patients and for clinicians and for all the stakeholders, the payers, et cetera. We keep saying it's non-sustainable. That have been an overread. It seems like we've adapted retina practices to be able to do these thousands and thousands of injections, but at some point it may become non-sustainable for sure. And even now, it's a real strain on the patients and their families, even for getting the cost. One final question on this before we move on to your current, digs at InflammX. Do you foresee realistically in office ever losing out to the OR anymore in this space of drug delivery? Could someone come up with a surgical device that would be so good and so safe that it would bring some of these diseases back into the OR that have now become mostly office-based?
I don't see it at this point. Anything that goes into the eye is a risk. And so whether it be OR-based or office-based, you're going in the eye, I think it's less invasive usually if it's in office. I mean, I think that's how I would distinguish between the two. If you have to go to the OR, it's probably going to be more invasive. And I think the risk is higher always for a more invasive product. So even if there may be benefits, and you've got to weigh up the risks and benefits for everything, of course. But I don't see it at this point. I do see a simple injected polymer carrying a drug to be a viable way to do this.
Yeah, I agree totally. All right, let's talk about InflammX. I want to hear a lot from you on this. And you and I have talked about it offline plenty. I'm a big fan of the concept. Why don't you just start with the basics? I don't think most of our viewers or listeners know much about this. What is InflammX? What is the product, the design of the protocol, et cetera, and even just the mechanism of action and the disease protocols you want to approach?
Sure. So InflammX was formed probably about two to 3 years ago. And the drug and the mechanism that we're targeting with the drug is the dysregulated inflammasome pathway of inflammation. So the inflammasome, or the NLRP3, is an innate immune system which exists in all of us, but it's relatively new. It was only discovered in 2002. And our scientist he spent his life in New Zealand, Colin Green, evaluating and researching this pathway of inflammation. And he was the assistant director of the ophthalmology department at the University of Auckland. So his first target was always looking at ophthalmology. And he developed a peptide in Auckland. And this was an intraocular peptide, which inhibited the inflammasome. So when this inflammasome was out of control and it becoming auto-inflammatory, he found that if he could shut it down with the peptide, that the tissue started to become physiological again. And so he then went further into how was it doing this? What is the mechanism? And that is the key to the technology and the mechanism. So what happens in disease is, obviously, if you have a disease or you get an injury or you get a virus or whatever, the immune system is activated. And in this case, the inflammasome for specific diseases is activated. And he figured out how it is activated, at least one of the mechanisms that it's activated. And this one is very upstream from what anybody else had been doing. So when I say upstream, I'm talking about at the cellular level. So what he found was that there's a hemichannel called the connexin 43 hemichannel, which is a protein, which is basically absolutely essential for life. Because what the connexin 43 hemichannel does is it sits in the cell membrane and it's closed, and it only opens when it docks with another hemichannel on an adjoining cell, at which point both of them open up to create what we know of as the gap junction. And that's the physiological function of the hemichannel. It doesn't do anything else. It opens, joins with another one, opens the channel, creates the gap junction, and two cells now communicate. When things go wrong with certain disease states, and we believe ophthalmology is a prime one, but there are others, particularly neurodegenerative diseases, where the connexin 43 protein becomes overexpressed. And so we get a lot of them sitting in the cell membrane, and they open up before they dock with an adjoining hemichannel on an adjoining cell. So now they're in the membrane, they open up, and ATP, which should be confined to the cell, gets into the extracellular space. When that ATP gets out there, that is a signal to the NLRP3 inflammasome to activate. It's a foreign body that it's signaling, and it activates. And as long as we have these membranes, the hemichannels, sitting in the membranes and opening prematurely, this inflammatory pathway is going to continue to be perpetuated. So we have a perpetuated inflammation going on in the body, particularly at those sites that have been affected. And this will go on and on and on until we can actually shut it down. Now, there's two ways one can shut this down. One can try and go into the inflammasome and specifically try to close the inflammasome itself. But that is a very complex assembly, and trying to do that will lead to off-target effects, which we have seen with certain drugs that had been developed. There was one called MCC 950 that Pfizer developed and had serious liver toxicity with the drug, so they eventually abandoned it. What Colin found was that we could, with his peptide, he could inhibit the inflammasome by closing the connexin 43 hemichannel. So he developed the peptide that would be this was for intravitreal injection. In an animal model of inflammation, he would inject it, and what he would show is that he could inhibit the inflammation. So then he started looking at, trying to find the real mechanism, and could he actually create ATP release, the inflammasome being activated, and us shutting it down with either the peptide or actually maybe with a drug. And so what he did was these very, very elegant studies showing that we could actually challenge cells, retinal pigment epithelial cells, get them to release ATP out of the cell. So this was pathological of the challenge. And then you can actually measure the activation of the inflammasome. So you can do a spot count, tells you how many spots, tells you how active that inflammasome is. He then challenged this whole system with the peptide and then also with our drug, which is called Xifalan. I'll talk about that in a minute. And what that does is very specifically works on an extracytoplasmic loop in the cell. And when the drug attaches to that particular loop, it closes the hemichannel. So it doesn't do anything to the hemichannel that's already docked and open because the loop's not facing the drug. The drug can only access the pathological connexin 43 hemichannels that are now eluding ATP into the extracellular space. So by doing that, we actually stop the activation of the inflammasome. And so we've got multiple animal models that we've done in terms of doing this with the peptide, but then we found this drug that GSK had developed 25 years ago. And as I mentioned, the inflammasome was only discovered in 2002, and GSK knew nothing about the inflammasome, but they developed a drug for migraine. And they ran pretty much almost 2,000 patients in phase 1-2 studies up to 18 months, showing the drug to be pretty safe and well tolerated. Side effects typically dissipated within a 2-week period. Dropouts were really low. And it worked in one of the trials, it didn't work in the other. It was the time that there was a merger between SmithKline and Glaxo, and this was one of the drugs that got booted onto the shelf. And then Colin found it through looking at patents, and this one had expired, of course, and looking at these patents and saw, wow, this looks like a hemichannel blocker. So he started to look at it. We licensed the drug from GSK, and then repeated all the experiments with the drug, and then ultimately licensed the whole thing from them so we could submit the IND to the FDA. So we got that done probably about 18 months ago, submitted the IND, which was a real interesting interaction with the FDA because this was still paper. All of the talks, all of the clinical stuff, everything was paper, nothing was electronic. Yeah. And anyway, we submitted it and they accepted it. And we then decided, okay, we're going to do a different thing from what anybody else is doing. We're going to look at indications in ophthalmology with an oral drug. We're not going to use the peptide. And so we're going into the clinic now with the oral drug. So along the way, we got the IND filed. We also put protocols together for DME and for intermediate AMD. Those were going to be our targets. The DME study got picked up by the National Eye Institute, the DRCR, and they liked it and decided they will run the study after we met with them, went through all of the mechanisms and also the IND that we had submitted, the brochures, the safety profile of the drug, and so on. So they decided they would fund it and run the study, and that study is currently underway. There's approximately 45 patients now enrolled in the study. So this is DME.
For the DME indication, so you're in the clinical phase now, just for clarity, and you're running the study with the DRCR. They're sponsoring some or all of that. Just for the investors in the audience, that's a non-dilutive funding at this point for your company?
Correct.
Great.
Yeah.
And so, and then that's one clinical endpoint, and the other one you're about to tell us, I think, on the intermediate AMD.
Right. So the intermediate AMD was an interesting one. So first we were going to do geographic atrophy, and then Phil Rosenfeld said to me, why are you doing geographic atrophy? He said, there's going to be 2 drugs approved. We should move to a different, earlier form of the disease. He said, you've got an oral drug. And this was at his angiogenesis meeting when I was talking to him. And we decided, that makes all the sense in the world. Nobody's going to inject into early AMD. Patients have good vision, and the patients don't want the injection, and the docs don't want to give it. So an oral drug seems to make all the sense in the world. But everyone said to us, but there's no endpoint. You can try and run a trial. What are you going to measure? And then Phil and his group published a study, a 5-year natural history study on AMD, and that's been published in one of the ophthalmology journals. And that 5-year study actually showed that there is an early signal. So there's a biomarker called a hypertransmissibility defect, which is measured by en face imaging. And that hypertransmissibility defect, they showed, ultimately starts to basically more and more of them are forming as the disease progresses. Still in the early stage, but hasn't affected vision of the patient yet. And eventually they start to coalesce and ultimately become the geographic atrophy lesions that we see today. And the patients then start losing vision. So we decided what we'll do is approach the FDA with the hypertransmissibility defect as the biomarker, and that we would show that we could either prevent it from forming or stop it from progressing in a clinical trial, which we looked at Apellis and Iveric, decided we'll do it over 24 months. So my first conversation with Wiley Chambers was really interesting, because I said to him, what we're going to do is look at patients at baseline, look at them at 12 months, follow what everybody else has been doing. He said, well, I'm not accepting that. I said, why? So he said, because if you show me baseline in 12 months, that doesn't tell me that you've had any impact on progression. You just show me 2 time points. That could be a pure fluke. He said, what I want to see is a slope of progression between the placebo and the drug. He said, that shows me, at least in the first year, if I can see that, then I know this drug is probably going to be successful in the second year. So he said, the hypertransmissibility defect, that's fine. That's a good biomarker. It's, photoreceptor dropout. You can measure it. And, but I do want to see multiple time points within the 12 or 24 months. So we redid the protocol, and we submitted actually 2 protocols. We submitted one for geographic atrophy and one for intermediate AMD. Waited the 30 days, didn't hear anything, and we're ready to go. So what we're looking at are, these lesions, and if we can inhibit them or prevent them from forming in the first place, because we believe that there's an inflammatory response going on that is creating the problem. And I think if we look at the GA studies that were done on the drugs that were, approved to complement, that tells me absolutely that there's an inflammatory response going on. And I think the inflammasome is a significant player in this disease. So that's where we're going now.
Oh, I'm sorry. Is this the first time that you're aware of the FDA recognizing this endpoint that you were just referring to, the hypertransmissibility findings, the lesions themselves?
Yeah, because everyone had told us they won't accept it.
But they did.
Well, they accepted it for us. But even more than that, there've been 2 retina meetings now where Wiley was at the meetings that this question came up because we had presented the hypertransmissibility defect as being a biomarker, and all Wiley was concerned about is, can you show me that you can show lack of progression? That's really the endpoint. But you have to have a biomarker that you're going to look at that's going to, be viable. And he said to us, okay. But then at 2 meetings, people got up and said, so you're going to accept that? He said, yes, twice. And then Phil spoke to him independently afterwards and said, are you sure about this? He said, absolutely. He said, I like it.
That's great. That's great news for all of us to hear. One final question on the protocols. When do you expect the first or next clinical readouts for either of these programs? It sounds like the DME program, the diabetic retinopathy program, is further along, and I know you don't have a crystal ball, but what is your anticipation for clinical readouts?
Yeah, I anticipate the DME will probably be towards the end of probably Q4 2025. And if we get the funding done relatively soon, we're raising funds now for the intermediate AMD. If we can get that funded, we will have a 1-year interim analysis, and that should read out around Q1 26. But that means we get the funding, this quarter or next quarter to get started.
So you're raising now to build out and execute the dry AMD program.
Correct.
Are you already fully funded to finish the DME program as it reads right now?
Oh, yeah. Well, the DRC are...
They're taking the whole thing.
They take the whole thing. We give them we have to give them some money in kind, but compared to what the trial costs, this is really, really a good deal for us. And I think the fact that they wanted to do it, I think, is also a really good sign for us as well.
Well, it's a big validation. This is a group that has done countless dozens of protocols in diabetic retinopathy. So good for you. And that's a validation. I want to thank you for coming on, Brian, and answering all these questions and walking us through what has been an amazing career, but also this new project with InflammX is fascinating. I have personal interest in it, as you know, and I want to, invite you to join us in Seattle and invite the audience to join us May 4th for OIS Retina. You'll probably hear from Brian there because he participates in our program routinely. Please join us in Seattle May 4th. Brian, you as well. Thank you.
I look forward to it, and thank you for this opportunity. Thanks, Feroz.